Thank you. Thank you very much. It's great to be back for a second action-packed day. And as usual, Dr. Van Devender queued up some really important topics. And I will just add a little bit to that discussion earlier that, it is really important for you to understand, and I'm talking just on a general level. I'm taking my FDA hat off to just describe some of the regulations that have happened over the years associated with nutrient management. EPA does have some, and we talked, yesterday, Dr. Van Devender talked about some of the local and federal regulations associated with nutrient management. And I was just reminded that, EPA does have some over level, not FDA, this is EPA, does have some overarching requirements for land disposal of waste, and manure is now considered a waste product. And I would go out on a limb to say that it is a potentially hazardous waste product. And the EPA has fined pretty heavily, I might add, some operations that are using their land as a disposal mechanism for manure. And I believe some of the fines went to some, maybe, I can't remember, but I believe there was a swine operation. Think about, massive hog lagoons out in, say, the Carolinas, for example, on hog capos, where, they land applied this wonderful resource to land. And it was determined by EPA that the land crops that were grown in that field, the nutrient demand for those crops was not sufficient enough to meet the amount of manure that they were applying. So basically, they're applying more manure, nutrient-wise, NPK-wise, than the crop that was planted could eat. So what happens with all the excess nutrient, and I love Dr. Van Devender's cartoon of that bathtub. Think about that spigot that's leaking all the nutrients out into the environment. That's what's happening when you apply more nutrients than your crop can use. And in particular, in our location where we are located in is a very heavily protected area. The Chesapeake Bay region is a very highly protected resource. And guess what? We have a lot of poultry operations in the Delmarva Peninsula, that's Delaware, Maryland, Virginia. It's one of the largest poultry operations in the country. So there are some very high regulations about what can be applied when and how often based on phosphorus and nitrogen content. So this is not FDA regulations, this is EPA. What I'm going to talk to you a little bit about is the food safety aspects of nutrient management. I did put a link in the chat box about a recent paper. This is a publication that came out of Dr. Danny Karp's lab out in UC Davis, and in conjunction with USDA, our Agricultural Research Service. This is something that I've been looking for a long time. This is hopefully this is just the start of a whole line of research that is piecing together how soil health and the maintenance of soil health can adversely affect any human pathogens that might come in contact with those soil particles over time. So this is sort of marrying a couple concepts of nutrient management, what you apply to soil, how often you apply it, and what you plant in that crop, what you plant in that field. So soil health, I don't think I'm smart enough either to convey what soil health is. I think the basic definition really is about, how nutritious is your vegetable that you're planting. That's it. That's if you have a healthy soil, you have a healthy crop. Well, we're learning that there's a lot of microbiology involved, and this paper I just sent to you is one of those tip of the iceberg publications that I think is opening the eyes of a lot of people, because the soil food health, soil food web is a very important concept. There's also a link in the chat box to Dr. Elaine Ingram's, who was, by the way, was a former CFO, the Chief Scientific Officer of Rodale Institute. She's done a lot of groundbreaking, really good discussion. She's had a lot of good discussions about soil health, what it means, how to maintain it. She and I don't see eye to eye, by the way, on some concepts, such as agricultural teas, and we can talk about that later. But in terms of soil health, she does have a lot of good points. So with that, sorry, I waxed poetic. I'll put my FDA hat back on. I'd like to talk a little bit about what we're doing and how we're doing it. And so we're doing a massive risk assessment. Yesterday, I put up a huge chart here, and I walked through it really quickly. I'm not going to walk through the whole thing again. I'll just tell you that there are 3 swimming lanes. Again, for those of you who are just tuning in, first swimming lane is understanding whether or not you comply. You need to comply with subpart F, and that depends on whether or not you're using a soil amendment of animal origin. Tune in yesterday's talk to help you figure that out. I think most of you are aware what an animal is, and if you're using any byproduct of an animal, you're covered under subpart F. So you need to go to swim lane 2, which is all about, are you making a treated soil amendment? Are you using a treated soil amendment of animal origin? And what that means is, has it been processed to either reduce the pathogens, potential pathogens in that product, or have you processed that product to eliminate pathogens in that product? So there's 2 different treatment options here. There's an injury level, reduction level treatment, or a elimination level treatment, and that will kick into either this triangle here, and I'll zoom in on that in a minute. This is 112.54A, which is our nuclear bomb approach treatment level. If you're using a soil amendment of animal origin with this level of treatment, and you have proper records, then you can go. And you'll notice I have the application methods are all in this section here. And I'm going to present this in a couple different ways because everybody learns a little bit differently. I'm going to circle, probably should have picked a different color, but there's different rows here. What this is what I'm referring to is during application and after application, right? So if you have a soil amendment that has absolutely no pathogens in it, it's been documented to go through a process that has to eliminate all pathogens in every single particle of that product, and you apply that, FDA has no restrictions. We have no days to harvest restrictions. You can broadcast this material. You don't have to pay attention really to how you apply it. If your baby spinach, for example, is looking a little chlorotic, baby spinach is a quick growing crop. You have a few days to harvest. You want to make sure you green it up really well. You can use a heat treated poultry pellet that meets this definition. Yeah, you're going to apply it in baby spinach is a low growing crop. You're going to risk, there is definitely potential for contact with that crop, but we have no restrictions on an application. Again, there's no restrictions after application either. So during application and after application, there's no restrictions on days to harvest. There's 0 days to harvest restrictions on this product. And here is the appropriate codified section that refers to that. We have no intention of changing this provision. We have no reason to. Now, your other treatment level, again, we're talking about something that has been treated to 112.54 Bravo. Okay, this is our second. This is a level which we would expect pathogens to be significantly reduced. This is, you're going to have residual pathogens in it, but they are very, very low levels, maybe even below detection. We do not require testing of your windrow, of your compost, regardless of your method of production, as long as you have time and temperature records that you meet the time and temperature necessary. We will accept those records as sufficient that your pathogens have been reduced to a level with which FDA deems acceptable when you apply them in accordance, which means you have compost, you have proper records, and let's say you have a row crop that, again, is looking a little chlorotic, and you need to apply some kind of nitrogen to it. Compost you have on hand. You can apply compost to that low-growing crop, even if there is a potential for contact with that crop. Okay, but you're minimizing the potential for contact. What does that mean? That means you're not blasting compost across your bed of leafy greens. You are carefully targeting the application, and that speaks a lot to application methods. There are many different ways to apply compost. Some operations have fancy machinery. Some growers have shovels, and then we saw a video of one such grower yesterday that does this. So as long as you're applying compost in a way that minimizes contact during application, this means that you have an established crop in your field, already established, the harvestable portion is present. You are allowed to apply compost, but you need to be careful about how you apply it. And we want to make sure that you need to apply it in a way that minimizes contact with that crop. So there's a lot of, we hope to come out with more guidance in the future that helps describe what this means, but it's hard. It's a tough concept, and we get that. Again, we have no intention of changing that concept. So the same applies for after application. Let's say you do not have a crop in that field yet. You're applying compost across that field. You're preparing your field to grow a crop. We have no days to harvest restriction. So you can apply your compost, either surface applied, till it in. There's no days to harvest restriction. You can plant your crop and then harvest it the next day. I don't know a crop that grows that fast, but there's no restrictions on that. And here's the appropriate codified as well. So that's basically it. You either treat it to a level that has no restriction on the application, or you're going to have a treatment level that minimizes. There is no other treatment level. The only other treatment level is no treated. And here we go back to untreated. All right, if we follow the untreated line down here, and I got a curve around here, and again, there's a little caveat. I don't know if you can read it. I'll read it to you. It says, untreated biological soil movements of animal origin except for human waste for 112.53, right? So if you're going to use human waste, you need to make sure it meets EPA's 503 regulations, and that's for a Class A biosolid, which would meet our nuclear bomb approach treatment level. So I had to make sure that was clear. We're not talking about human waste here, but animal waste you can use. And if we're using animal waste, any animal waste that's untreated, and I will loop in agricultural teas here, because pretty much every agricultural tea that I've seen lately, I would classify as an untreated. Okay, so how do you apply your ag tea? Well, you need to make sure you're applying a tea. All right, let's say during application, you have an established crop in your field, you're applying agricultural tea. You need to make sure there's no contact. An untreated ag tea, if your tea is untreated or you're using raw manure, you need to make sure that there's no manure getting in contact with your harvestable portion of your commodity. I think that makes mostly common sense by now. Keep your poo off your produce. I think that's now codified. It applies sort of some teeth to that good agricultural practice. So you can't do that. All right. So what about if you have a field that has no crops in it, right? You have a fallow field. You want to prepare it for the next crop. You need add some nutrients to it. And we're learning that more and more that raw manure is an important component of healthy soil systems. In a lot of regions, the growers that use raw manure, we're not preventing the use. We just want to help growers use it safely. I can tell you, based on the previous talk we just heard, our research has shown a lot that, compost doesn't have as much nutrients compared to raw manure, but it has a stabilized form of nutrients, which will help when you're trying to keep the nutrients where the crops need them to be. Compost is, in a lot of ways, it's a much better option, certainly for a food safety option, but compost certainly can be used in your targeted approach to maintaining food safety. And the publication I included in the chat box speaks to the benefits, the microbiological benefits of compost in terms of reducing any human pathogens that might creep into that. So if you're using raw manure to amend your soils, you cannot have, all right, so during application, you cannot have any contact. I think that's clear. And if by chance you have no contact after application, let's just say an example would be a fruit tree, for example, an apple orchard, and you want to take a shovel and apply raw manure to the base of a fruit tree. Now you have 0 days to harvest. You can still harvest those apples because there's no contact during application, no contact after application, right? You're not blasting manure into that orchard. You are not, there's no contact with the apples at all. Now, there's some provisions in the produce safety rule that does question this practice, so it might not be the best example because, yeah, you have ladders of workers, boots going into the manure, and then you're going to have poopy ladder rungs, and hands are going on the ladder rungs and picking apples. All right, so there are rules against drops as well. You cannot harvest an apple once it dropped into that manure or any soil for that matter. So that's just an illustration of what we mean here. If there's no contact with the commodity during application, no contact after application, there's no days to harvest restrictions. So the last thing is where all the magic is happening right now, all right? So we're talking about how most growers use manure. Most growers use raw manure or any untreated soil amendment to modify amend their fields, their soils, to get the nutrients where you need them to be for the crop, to meet the nutrient demand of the crop. And that means you're applying that manure in a way that minimizes contact after application. There's no produce in that field during application, so you can apply it any way you want. No big deal. There's no contact with the produce. Now, again, be aware of what's around your field. If you got a hammer truck and you're hammering this, this raw manure, or you're spraying the raw manure with a boom sprayer, what about wind drift? Do you have a crop downwind? That's also covered in the rule. You need to pay attention to those practices. But if you're being careful and there's no contact, no problem. So what does this mean after you apply it? How soon can I plant my crops? What's the days to harvest restriction? What does FDA think about that? Well, we say you can do this practice because you're applying raw manure in a way that minimizes contact with your crop. You're minimizing the potential for contact with your crop. If you plant that crop in the soil after you apply raw manure, plants got to grow. The commodities, it's a stem fruit or low growing leafy vegetable. Yeah, if you applied it in a way that minimizes the potential for contact after application, that meets this definition. All right. So here's what we're working on. We're working on this. This is already in the USDA National Organic Programs rule. It's a 90 to 120 day rule, depending on how you're growing your crop. This is guidance. This is not a rule. This is guidance. If you want to be an organic grower and you want that USDA NOP seal on your produce, so you can command that high value when you sell it, you need to pay attention to 90 to 120 days. This concept was not born out of a food safety concept. If you look at the NOP's guidelines and rules and regs, look on their website, they're going to say this is no bearing on food safety. And once FDA started to get in the food safety game on the produce level, USDA decided that they will punt to us when we figure this out. So what we did was we looked at the data, and we've been looking at the data for several years, and we pumped millions of dollars into it to figure this out. That's what I'm here to talk to you about today. Again, I've got more slides than I'm going to talk about, but I'm putting them up here so you can look at this recording, pause it, take a look, and say, okay, It's just another way of looking at the data. This is the entire subpart F into one chart as well. You've got treatment level. This is all treated, right? You've got nuclear bomb approach, treatment A, no detect for any pathogens. You can apply it in any manner, no restrictions, 0 days to harvest. Compost fits into this lesser treatment. So it's a magnificent way of reducing pathogens, a very simple way to do it. You can do it on your farm. We saw a great example yesterday. Yeah, it has the potential for pathogens in it as long as you apply it in a way that minimizes potential for contact after application and during application. We have 0 days to harvest restrictions. And here are the records you need to keep. If you're going to make your own compost and apply it, if you're going to side dress your crop with it, you need to make sure you show us these records that the compost has been adequately treated. So everyone asks this, and I think it's an eye opener for most people that we expect pathogens in the compost. Yeah, we do. And this is green waste compost. This is a great study, by the way. I recommend you look it up. Will Brinton runs an institute up in Maine. He's kind of I regard as a compost guru. He runs a school, Maine Composting School, and he did a study back in the day. This is decades ago, but I use this as an example because it's a study based on commercial compost that we do not regulate under subpart F. This is green waste compost. So this is municipal, trimmings, leaf yard waste that is being produced into soil amendment through thermophilic composting. And 1% of all the samples that he found had salmonella, and over almost 30% of the samples he found had fecal coliform content greater than 1,000. Now we could have a whole debate on what fecal coliform means, because this is one thing where Dr. Elaine Ingram and I don't see eye to eye on what E. coli and fecal coliform means in terms of public health. But E. coli, ONFS 787, this is interesting because these composts were pulled, this is in the Pacific Northwest, these composts were pulled about the same time we had the 2006 baby spinach outbreak in Salinas. And we're not sure if any of this compost were used for production, but the Leafy Green marketing agreement, basically was a whole industry born out of this outbreak. So unfortunately, we don't have these isolates to match whole genome sequencing, but I found this paper very interesting because this involves a green waste compost. And if you're not using a soil amendment of animal origin, you're probably using a green waste based soil amendment. And, nothing is without risk. And this is why. So these are just some examples of commercial operations that we've been working with. Again, this is another way of presenting the information I just said. If you're using a raw manure and you have no contact after application, then there's 0 days to harvest interval with raw manure. There's no if you have no contact during and no contact after, there's no contact, no problem, no days to harvest. But we're talking here about filling in this reserve portion. So now you're applying manure in a way that minimizes the potential for contact. You've amended your soils with manure. How long do you have to wait? And this is what we've been trying to figure out. And we've learned a heck of a lot. Okay, so we're doing this risk assessment. We spent millions of dollars. We've figured out on a national level on the East Coast and West Coast, we've had a huge program spearheaded by the Western Center for Food Safety. Basically, we wanted to know how many pathogens can we expect in raw manure. And so we just went ahead and sampled as much manure and manure-based compost that we could, raw manure, really, mostly to understand the persistence of manure in compost. Raw manure. Persistence is the prevalence of pathogens in raw manure. And that was chapter 1. Chapter 2 was trying to figure out just how long those pathogens might persist in soils over time. So if you have raw manure, the pathogens of raw manure, and you're applying it to your field, when can you expect it to die off? in FDA's eyes, where is that risk level? And again, NOP has 120 days. Fits right around here. This is just a crude graph to just illustrate the die-off curve, pathogens, when can it be expected to reach a level that FDA is comfortable with planting crops or harvesting crops for that matter. So this is probably the technically most, and we've done this, by the way. We've done step 1, we've done step 2, and now we're on really step 3, which is, okay, you've applied manure to your field. We know how much pathogens are in there based on our survey. Depending on how much manure you apply will, of course, depend on your crop. We learned from Kurt that you need to understand what your crop needs, and that will ultimately determine how much manure or any soil amendment you need to add to meet that nutrient demand. So that's an important play here. But what we're talking about here is transfer coefficients. Okay, let's say you have residual pathogens in that crop when it's ready to harvest. Let's say you had a rainfall. Let's say you have overhead irrigation event. And we're talking here about the potential for splash. And this is a step number 3. We've got 3 types of crop we're looking at. We're looking at crops that grow underground, root crops, and this is, carrots and onions. We're looking at crops that are low growing to the ground, like leafy vegetables, and stem fruits, like, tomatoes are good examples. So this is the really transfer coefficients and we need help with, and I implore any of you scientists out there working on the bench to try to, send us as much data as you possibly can. We're working hard on this ourselves, but we need help. And then step 4, of course, is, okay, what happens? What happens to that commodity once it's harvested? It goes through the cold chain storage, it goes to retail. Sometimes that means it's temperature abused in the open retail display cases in your refrigerators and in the supermarkets. But most of the contamination that we know happens in your own homes. Okay, so we're talking cross-contamination with your cutting boards, your knives, the other food products that are raw, like chicken or beef, ground beef, food safety practices, depending on your knowledge and practices at home, will depend on whether or not you get sick, and in many cases that's the case. So this is where FDA stops. We'll never go into your houses. We sort of draw the line there. We will produce, communication pieces and education on, how to store your food safely. Do you need to wash your produce? That sort of thing. But we'll never create a rule that will impinge on how you eat your produce at home. But this is a factor, and we already know all this. So I'm just going to rip through these slides because I want you to pause. Chapter 1 was what we did, and who the players were, and hats off to these guys. They really worked hard for us. They looked at poultry litter, dairy manure. We dug piles for years. And I want you to pause the slide now when you get this recording to look up these papers. These are the papers that have come out to date that illustrate and highlight the data. And interestingly enough, the pathogen prevalence in manure, salmonella in poultry litter, E. coli in dairy manure or beef manure, they're all the same on the West Coast and East Coast. The numbers were about the same, which was good because it tells us that we have less variability. We've done a lot of regulatory samples in compost. This is a compost out in one of our investigations. We found pathogens in it. I will fill you in on those details. But I really want you to look up this paper. Dr. Sharma, there's 2 papers I need you to look up that highlights our model. Dr. Sharma's paper, if you can, grab this, I can send it to you, send me an email, and I'll send this to you. It's fascinating. This is the largest study ever done in the history of soil amendments. Okay, we have more data points here that look at exactly what we need to populate our model, and our model is this. So this is the second paper I need you to look up. Dr. Peng, he's sort of the wizard behind the curtain here at FDA, our modeler who has designed for the first time a predictive model for how E. coli, O157, and generic can persist over time in soils. And kind of what we found is not going to be any surprise. This is a chart from USDA's paper. You can see that these are the main factors. And what we found was, and this ties into the nutrient management. Okay, how much manure you're adding really was the main. These are not the only factors. These are the main factors that determine how long pathogens survive. Okay, so initial inoculation level, poultry litter seemed to hold on to the populations the most. And it's just, there's so much variability in here that our data set teased out. It's just an absolutely fascinating paper. The third paper, series of papers that we're just coming out with now illustrates transfer coefficients, because that's where, that's the most important thing. If the pathogens are not getting onto your produce, there's really no concern. The more investigations we do, the more we find pathogens in soils. And you know what? I say, no big deal. I'm not surprised when I find a pathogen in the soil. What I don't want is that pathogen to get on a produce that goes to market. And there's a big difference in our response. So transfer coefficients, this is one paper that came out to illustrate our first paper that came out, and this talks about transfer of soil pathogens from soil to cucumbers. And it's complicated. It's really complicated. This is just an example of all of our data for 2018. We have a lot of transfer. Well, I'd say a lot of transfer. Well, it's not a lot of transfer, but we had transfer from soil to produce in 2018. But the same study in 2019 didn't see any. So there's a lot of differences in seasonality here that we're trying to get a handle on, and soil type as well. So I can't remember how long I'm supposed to be talking here, but I'll keep going until you make me stop.
Dr. Ingram, we do have a question if you want to pause real quick. Okay, so the question is the risk higher in different amendments, goat, sheep, cow, or horse, or are the risks the same?
The risks are, okay, so the level of pathogen population in manure, so salmonella, so it depends on what pathogens you're talking about. Salmonella is typically associated with poultry litter, poultry manure. Typically, you're going to have E. coli as well, but to a lesser extent that we're finding. And E. coli, the host is typically cattle. And so the difference is in the nutrient content of those manures. And Dr. Van Devender will tell you that, poultry litter, poultry manure has a much higher nitrogen content and phosphorus content than other manures. So that makes a difference, because what we learned yesterday is that, you're not just feeding the microbial populations in the soils, you're feeding potential human pathogens as well with your chemical fertilizers, with your, the available nutrients from your manure fertilizers. And because that poultry litter, and this is all conjecture here, but we're pretty sure that because poultry manure has such a higher nitrogen content, A, you don't need as much to amend your crops, but B, it's got a lot more available nutrients for pathogens in the soil. And this is a potential concern for us, because we give heat-treated poultry pellets, for example, a free pass. We say, go ahead and apply this stuff in any way you want. There's no days to harvest restrictions. Go ahead, use it willy-nilly. If your crop needs nitrogen, we prefer you use a treated form of soil amendments like heat-treated poultry pellets, and there's less risk. However, if you have any residual pathogens in your soil, from a bird dropping, from a feral swine came through and took a dump between your rows, now you're applying heat-treated poultry pellets to it. And guess what? You're enriching that human pathogen population, and now you're going to have greater potential for splash. So in all of our studies, we're looking at poultry litter as an exponential blossom of human pathogens, and pathogens in soils can maintain their population size for longer when poultry litter is used. So I'm not saying that, you know, I need to be a little cautious because poultry litter can be used absolutely safely. And we encourage, you know, organic amendments like poultry litter to be used. We much prefer organic amendments to be used than chemical fertilizers from a soil health standpoint. But you need to, you know, just to answer the question, yeah, we're looking at poultry litter with a new lens. So our survival curves, you know, Dr. Peng had a huge task. We had so many data points. And these are all the survival curves. And if you've never seen stuff before, I'll just gloss it over because you're seeing a slope here. In every one of these, yeah, this is a population, you know, the population of bacteria. Sometimes they blossom. Sometimes they grow exponentially, you know, when you have raw manure applied to soil. Sometimes we have a lot more growth, you know, on day 2 or day 3. But by day 14, guess what? All the populations seem to decline. Sometimes they grow and then they decline, they grow and then they decline. Sometimes they're like a roller coaster here. They sort of go up and down, but guess what? They all go down. And that's great news for growers that want to use raw manure because the population is declining in every single scenario. And what Dr. Peng did was took a model called random forest model. Here's a slide that illustrates what that is. It's all rocket science to me. I don't understand any of it, to be quite honest with you, other than for the first time, we're able to model all those data points under one umbrella, and we were able to validate it. So we used our data set to train the model. We used 30% of the data to test the model, and guess what? It absolutely works. This is sort of regression curves. Most people are familiar with the regression curves, but if you know, the closer to 1, the better. And 0.89 is pretty close to 1, which means, yeah, I think we can safely say our model is definitely validated against the raw data that we've seen. So we're able to model human pathogen persistence and prevalence in soils over time. And, you know, I've been trying to do this for years on the bench, never could do it. And now finally we have something. But I will say this, we're looking for partners because we're able to do this on the East Coast. We're only able to study 3 geographically different locations on the East Coast. We need partners on the West Coast as well to do a similar study to make sure that our model works out on we have no reason to believe it won't, but it would be helpful.
Dr. Ingram.
So, yes.
We have a few more minutes before we need to wrap up.
Great. I'm just going to walk through. Well, I'm done. Basically, I'm done. I just wanted to highlight the model, that it works, give you the papers to look up, and that's basically it. I want you to know that we're working hard to figure out, you know, what raw manure means for a grower in terms of food safety. It's still very much, we're several years away from proposing any changes to our previously, you know, reserved portion of the rule. We're going to reopen Subpart F in about 5 or 6 years to help understand what that means. And nothing will be a surprise. You will all have a chance to comment on it, and then we'll bring it back and then reassess and reevaluate and go through a whole, you know, comment period. And there'll be a huge educational piece to this as well. So I thank you for your time.